But welcome everyone. My name's Heather Glon. I'm the lobster research biologist at DMR, if we have not met yet. But it is a pleasure to be talking with you all about a study that's been going on since 2023 with a fantastic team, talking about molt increments and predictive indicators within the Gulf of Maine on lobsters. So just to start out, I like to start with acknowledgement sometimes because this project is truly a team effort. It's made up of a lot of DMR staff, contractors, industry members from Inland Seafood, Ready Seafood, and Bold Coast, and then also academic members from Maine, and then a lot of folks from DMR have been on this as an advisory capacity. So just wanted to put that out there as these are the folks who have done a lot of the physical work for this, and I'm just kind of pulling everything together. Just so we're on the same page in terms of lobster growth terms, I'll be using CL for carapace length. Molt interval is the length of time between the molting events, and then molt increment is the increase in size after molt. And so all of these things can be variable based on size, sex, reproduction, environment, genetics, and a whole host of other things. In terms of looking at a lobster and deciding whether it will molt soon or not, we've got some external characteristics. We're also going to be talking about the BRICS index, which is taken by taking the hemolymph, and that measures the total protein concentration within the hemolymph, and it is used sometimes by industry members to kind of look at health and then also how close they are to molting. Also talk about cetal staging, which is the visible separation of the new from the old shell within the pleopods. And that's most easily seen underneath a microscope, but can be also done without harming the animal as well. And then if you didn't see Emma Dillart's poster yesterday, I think it's somewhere in the room or somewhere else, but definitely check her poster for a new method that we're working on to look at molt probability. Just a bit of a background, and I am not going to go into detail on this, and instead I'm just going to point you to two other talks, Tracy Pugh's and Jenny Nestledge's later today, talking about the lobster stock assessment review, as this was a research priority identified within the stock assessment, and then also Jenny's talk on the seasonal growth model, which is going to be really interesting as well. But we're hoping to bolster that data set that goes into both of those items and eventually the stock assessment and fill in some of those data gaps there. So that's kind of the root of where this study came from. So our main objective, like I said, is to update those growth data sets and model after Massachusetts DMF work. So Tracy in the back is a co-author, and they really set the groundwork for a lot of the methods that we applied during this study. And we are hoping to describe the molt increment, document timing, seasonality, and impacts of temperature on time to molt, and then also compare the common molt indicators. And just so that you're all aware with the timeline, 2023 was a pilot study, figuring out the method and how things worked. And 2024 and 2025 is the full data collection. We still have lobsters in enclosures, so this is not a complete data set. So our methods, we supplied all of our lobsters from Ready Seafood, Bold Coast Seafood, or the DMR monitoring surveys for ventless trap surveys or sea sampling. Looked for potential candidates, brought them back to the lab using this amazing data intake sheet that my first year contractor, Elizabeth Camp, put together, and basically assessed them all and then kept the highly likely to molt candidates. We acclimated them for 24 hours because sometimes they were kept really cold at Ready Seafood. And then we held them in enclosures, waited until they molted, and then measured and released them. So it was a really elegant way to get a very straightforward data set. Inland Seafood Lobster Pound was our study site for the first 2 years. Unfortunately, they are no longer under operation. So in 2025, we moved to DMR's docks, much closer, much easier to access, which is great because Emma was working on a study that required her to do full sampling every Wednesday. So this actually worked out great. A rare photo of me actually pulling up the enclosures is included. So our final data set is 134 individuals. We looked at over, I think, 450 individuals at the lab, and 134 were able to molt, and with under 2 months. Just a quick size distribution, and these size bins are based off of the stock assessment. So definitely skewed towards the smaller individuals, which are still above legal size. We are including V notches in this as well, but females and males. We still have a lot of females in the enclosures. I'll tell you why in a minute. And in terms of statistical areas, we tried to get even representation across the geographic area as well. Still working on some of those bins as well. So in terms of our results, I'm going to focus on increment timing and indicator effectiveness. So just keep those three areas in mind. Working first from increment, looking at the male and the female. Males and females, unsurprisingly, they have significantly different molt increments. So therefore, throughout the rest of the analyses, I'm keeping males and females separate. So about 10.02 millimeters for all the males, and for the females, about 8. Looking at the statistical areas, there's no significant difference between the molt increments in the different statistical areas, so I did combine them throughout the rest of the analyses. And the molt increment, so this graph on the bottom is the pre-molt carapace length, so what size they came in at, and then the molt increment is on the y-axis. And looking at males and females, females are pink throughout the rest of the presentation, males are blue. And the molt increment is independent of the pre-molt size for both sexes. We do have some larger females still in the enclosures, so those may add some more of a significant trend to that apparently negative slope. Also looked at percent growth with those same data. Large lobsters grew proportionally less, and the size effect that we were seeing is stronger in females than in males. Moving into timing. So this just shows all 3 years combined, May through November, and then just the count. Males molt earlier than females. And then I broke these down by year as well, and then as well as pre-molt carapace length on the y-axis. So the molt seems to be occurring later in 2025 compared with 2024. No surprises there. That's what industry has been chatting about as well. It's colder this year than last. So that's pretty consistent. And as you can see, we've only got through the beginning of October with these data, but we still are getting a lot of molts. Did a quick linear model to look at molt timing and size. And molt timing is more size dependent in females than in males. And so this was calculated out at about 2.4 days for females per 1 millimeter increase in pre-molt carapace length, something like that. No difference, no significant difference in male molt timing by carapace length. Moving into indicator effectiveness. So how do you tell when a lobster is about to molt? First, looking at external characteristics, and this brings us back to that data sheet that Elizabeth put together the first year. And this is based directly off of the Massachusetts DMF data, and so we do have comparable data in Massachusetts that we can look at together combined there. So we had about 447 individuals that we collected these data from and circled each of the none, light, moderate, and heavy for each of them. Came up with this graph. Do not look too closely at this. There are a lot of different categories in there. Main take home is there's just no reliable external characteristics that we recorded that can be used to predictably, to predict molt. We did find that the dark purple abdomen, which is kind of like you know it when you see it, and the blue maris are helpful, but not always completely accurate. Looking at bricks, difficult to predict during using bricks. And so the days to molt is on the bottom. Pink this time is the ones that have molted, and blue are the ones that did not molt. We didn't really find that this was a good predictor of molt, even though it is assumed to increase when they're preparing to molt until decline just before molting at that cetal stage of about 5.5 and just pre-molt. Looking at cetal stage, this was definitely the most reliable indicator. We got a pretty consistent trend downwards. So quicker days, the more they progress through the settle stage. So that's great. There's been a lot of past historical work decades and decades ago into this. So it's nice to know that this still holds true and that the way of measuring the settle stage is pretty, it's a learning process, but you get better at it. Thank you. So females definitely progress through their stages faster. Remember that they molted later in the season, so this makes sense. I'll make this super quick. This is just a quick plot that I put together at the end of last week. And looking at time to molt by settle stage and pulling out a 50%, so median days to molt based off of the proportion not yet molted and then moving through the days to molt on the bottom. So we were able to figure out days. Females seem to move quicker, which backs up the previous plot as well. Looking at holding temperatures, we did hold them in open water, so our temperature was variable throughout the season, but we do have temperatures, did use a HOBO logger on the pasture, we just haven't pulled and analyzed those data yet. But I did calculate the mean daily temperature for each of the individuals. Put together a mixed effects model, and so settle staging is reliable to predict the time to molt, so everything there makes biological sense. Temperature accelerates molting, so that's supported by the model. And then higher temperatures actually increase stage progression as well. So this I found really interesting. So under higher temperatures, the early stages of males, early stages meaning settle stage, are quickened more than the later stages according to temperature, whereas females have a uniform response to temperature across all the stages. So I'm looking forward to digging into this further because that's really interesting to me. Major take homes. And so under increment, so males, they molt more than their increment is larger than females, probably due to reproductive needs as well. Most of these, if not all of them, would have been reproductively mature of the females. The increment is independent of pre-molt size, but females may decrease as the pre-molt increases. Males molt before females in terms of timing, and the temperature does appear to accelerate that molting process as well. In terms of indicator effectiveness, I think settle staging is winning so far. That was kind of like to be expected, but it's really neat to be able to compare these different methods and then also kind of build the groundwork for Emma's work looking at hormones with the molting process as well. Next steps, I mean, first thing is to complete the season. It's getting cold, so we do want to get those enclosures out. Looking into more of the holding temperature effects because we do have variable temperature over the years. Got it. Last slide, Chris, don't worry.
I don't know where my timekeeper went.
Yeah.
I'm doing the job.
Perfect. And then analyzing like a spent agar status also. We did have some, especially last year, come in as spent agars and looking at how that might affect the time to molt will be really interesting and could lead the way into a future study as well. Jointly combining Massachusetts and Maine data as we collected very similar data streams on purpose so that we could make them comparative. And also discuss with the technical committee to figure out, and Jenny, of course, too, to figure out how to identify an impactful and relevant data set for their models. So, and Emma's going to continue working on the molting hormone side of this and molt probability. And so everything's kind of like snowballing at that point. But, all right, any questions? Eric.
Okay, come on, check. No. There we go. So you said that you need a microscope to do the settle staging. Is there a way to do it in the field or in situ that you can do it quicker and easier?
I'm so glad that you asked. I did not plant that question either. And so what we started doing, we can't get a fine scale settle stage in the field super easily. But we did get little LED lights and tested out a bunch of them to hold underneath the pleopod or hold them up to a light. And you can get pretty close, especially with the later stages. You won't be able to see the CD coming in super well, but you can see that line start to pull away from the tip of the pleopod, the new shell underneath. So you can get pretty close, but getting that like 0.5 kind of fine scale is best done in the lab or with a very good macroscope like camera or macro camera, sorry. Yeah, Gail.
Thank you. Great talk, Heather. Given that you have the hobo loggers and so you have fine scale resolution on temperature, have you thought about incorporating some kind of growing degree day into your predictions? I don't know how that works with like incremental growth compared to continuous growers, but it could be a really cool component.
That sounds great. Thanks. Can you write that down for me? No. No, I have been thinking about ways to kind of get more at that fine scale resolution rather than just mean daily temperature, because they're all held different lengths of time, sometimes a week, sometimes 2 months. And so I do want to get a better way of incorporating temperature in. So yes, thank you.
I understand that you're trying to estimate time to molt in the wild. However, is there any rationale for in your experiments to see whether amount of food availability would affect? Because in certain, my expertise in insects is that food availability controls molting.
So I think that's one thing that we're trying to reduce lab effects. So we are feeding them. So we're feeding them all the same amount. So we haven't looked at how food may impact those last stages of molting. I'm not sure if that's where your question was going. We haven't held them long enough to hopefully make an impact of food availability. We're just keeping food available for them.
Well, having kept some lobsters in captivity, I've seen that the more you feed them, the faster they molt, and so it would be interesting to see whether your like the cedal, your most accurate measure, the cedal measurement would be associated with feeding, and that would give some idea about looking at food availability in the environment as a control for the development. I mean,
that would be interesting.
Yeah.
Yeah, we can do a starvation experiment in the future.